Hi everyone, this is Anita from St. Joseph's College of Arts and Science, Autonomous Cuddalore. So today in this video we are going to see about the topic micropropagation. So micropropagation is an important technique and it is a complex process, and this is an overview of micropropagation. So before we go to the topic, we just see introduction about micropropagation. So generally, in case of plants, it can be propagated by two methods. One is sexual, another one is asexual. In case of sexual method, takes place through the generation of seeds, and in case of asexual method, takes place through the multiplication of vegetative parts. So next one is clonal propagation. It refers to the process of asexual reproduction by multiplication of genetically identical copies of individual plants. It has been successfully applied for propagation of apple, potato, tuberous and several ornamental plants. So there are two propagation. One is in vivo clonal propagation, another one is in vitro clonal propagation. In case of in vivo clonal propagation, the propagation is very tedious and it is expensive and it is frequently unsuccessful. But in case of in vitro clonal propagation, here the tissue culture is referred to as micropropagation, which is also called as micropropagation. So in the case of in vitro clonal propagation, here we will be using of tissue culture for micropropagation, and this was started by Morrell in the year 1960 for propagation of orchids, and it is now applied for several plants. And micropropagation is an handy technique for rapid multiplications of cells. Next one is techniques of micropropagation. So as I said earlier, micropropagation is a complex and process, and it involves 5 stages. Stage 0, that is selection of mother plant and its maintenance. Stage 1, initiation and establishment of culture. Stage 2, multiplications of shoot or rapid somatic embryo formation. Stage 3, in vitro germination of somatic embryos or rooting of shoots. Stage 4, transfer of plantlets to a greenhouse environment or natural environment. So these are all 5 stages involved in the micropropagation, and these are all steps involved in the micropropagation, that is selection of explants, surface sterilization, media selection, multiple shoot bud induction, shoot elongation, rooting and hardening, and plants in field. So there are different approaches can be followed in case of micropropagation. They are: first one is multiplication by axillary bud or apical shoot. Second one is multiplication by adventitious shoot. Third one is organogenesis, and fourth one is somatic embryogenesis. So first one is multiplication by axillary bud and apical shoot. Quiescent or actively dividing meristem are present in the axillary or apical shoot. This axillary bud located in the axils of leaves are capable of developing into the shoot. So by means of induced in vitro multiplication in micropropagation. So it is possible to develop into a new plant from meristem or shoot tip culture or from bud culture. That is, a new plant can be developed from the choosing of explant. It can be a meristem, it can be a shoot tip culture, or it can be a bud culture. Second one is meristem and shoot tip culture. So apical meristem is so while we are choosing an explant, mostly we'll be preferring of meristem. So while we are collecting the apical meristem, it is a dome of tissues. That is, in the tip of the shoot, there will be the tissues will be cut in the form of dome. So that is said to be dome of tissues. And the apical meristem along with the young leaf. If the apical meristem we are collecting along with the young leaf, primordia, then it is constitute the shoot apex. For the development of disease-free plants, meristem shoot tip should be cultured. Meristem or shoot tip is isolated from a stem by V-shaped cut. That is, while we are isolating any kind of meristem, it should be cut in the form of V shape. Size of the tip is critical for the culture. That is, frequently the size should be 0.2 to 0.5 mm. In general, the larger the explant, better the chances for survival. That is, the size of the explant should be larger so that the survival rate will be increased. And most widely used media for meristem culture are MS medium and white medium. And in case of stage 1, culture of meristem is explashed. Then addition of growth regulators, namely cytokinin, auxin, and will support the growth and development of the plant. In case of stage 2, shoot development along with axillary shoot proliferation occurs. High levels of cytokinin are required for this process. In case of stage 3, associate with rooting of shoots and further growth of plantlet. Root formation for root formation will be adding of low cytokinin and high auxin. And optical temperature should be around 20 to 28 degrees Celsius. Mostly 24 to 26 degrees Celsius should be maintained. Next one is bud culture. The plant bud possesses quiescent or active meristem depending upon the physiological status of the plant. So there are two types of bud culture can be performed. One is single node culture, another one is axillary bud culture. Single node culture, it is a natural method for vegetative propagation of plants both in case of in vivo and in vitro condition. A bud along with a piece of a stem is isolated and cultured to develop into the plantlet. Closed buds are used to reduce the chances of infection, and in case of single node culture, there is no need of using of cytokinin. Next one is axillary bud culture. So in this method, a shoot tip along with the axillary bud is isolated. The cultures are carried out with high cytokinin concentration. So as a result, apical dominance stops and axillary buds develop. For a good axillary bud culture, the cytokinin and auxin ratio should be around 10:1, and this will be variable and depend upon the nature of plant species. In general, juvenile plants requires less cytokinin when compared to that of adult plants. Multiplication by adventitious shoot. Stem and leaf structure that are naturally formed on the plant tissue in sites other than the normal leaf axil region are regarded as adventitious shoot. There are many adventitious shoot which include stem, bulbs, tubers, and then rhizomes. Adventitious shoots are useful for in vivo and in vitro clonal propagation. The meristematic region of adventitious shoot can be induced in a suitable medium to regenerate into the plants. So next we move on to factors affecting micropropagation. So there are different factors which may affect the micropropagation. First one is genotype of the plant. That is, we have to select the right genotype. And second one is plant with vigorous germination and branching capacity are suitable. So second factor is culture media. Standard plant tissue culture media should be used. Addition of growth regulators, that is auxin and cytokinin, and alteration in mineral composition should be takes place. Third one is physiological status of explants. So explants from more recently produced parts of plants are more effective than that of older regions. That is, newly produced region can be mostly used as an explant when compared to the older regions. Next one is cultural environment. So cultural environment is one of the important factors which affects the micropropagation. So first one is light. Photosynthetic pigment in culture tissues absorbs the light. The quality of light is also known as influence in vitro growth of the shoots. Example, blue light induced bud formation in case of tobacco shoots, and in general an illumination of 16 hours per day and 8 hours night are satisfactory. Next one is temperature. Optical temperature around 25 degrees Celsius is very much needed. Some exception is in case of begonia and in case of other hybrid tissues which may grow at low temperature of around 80 degrees Celsius. Next one is composition of gas phase. Unorganized growth of cells is genetically promoted by ethylene, oxygen, carbon dioxide, ethanol, and then acetaldehyde. So these are all overview of micropropagation. That is, micropropagation is a complex process and it is involves 5 stages. That is stage 0, stage 1, stage 2, stage 3 and then stage 4. That is, first step is choosing of one explant. That is, choosing the explant is very much important in case of micropropagation. And second one is inoculation. So second step in case of micropropagation is inoculation, and next one is callus culture. Then after callus culture, shoot regeneration takes place. Then after shoot development, root development takes place. So after root development, hardening of tissues takes place. Then at last we are transferring the plant to the natural environment. So this is the overview of micropropagation. So in case of micropropagation, it is a complex process and it is an important technique, and it has different approaches. That is, more than 4 to 5 different approaches can be applied in case of micropropagation. So main thing regarding micropropagation is choosing of an explant. That is, from where we are collecting the explant is very much important in case of the micropropagation, and there are different factors which may affect the micropropagation like temperature, pH, environment. So these are all some of the factors which may affect the micropropagation. So main thing regarding micropropagation is choosing of an explant. So where we are collecting the explant. So when we are collecting the explant from the older region, the result what we get is not very much good when compared to the plants that we are explant that we are choosing from the young plants. So that is an important thing. Second one is environment. Then some physical factors may also influence the micropropagation techniques.